I'm Emma Newcombe. I'm from the Cawthron Institute in Nelson, and what I'm talking to you about today is a report that my colleague Liv Johnston and I put together, completed this year, on what we know and what we can say about the state of environmental health in Picton Bays. So I'm talking to you today about quite a small area, Shakespeare Bay, Picton Harbour and Waikawa Bay. So as you are all very aware, these are important for a number of reasons: commercial, cultural, recreational, and that's on really both a regional and a national level. I'm talking to you today mostly about information from a Western science perspective. Obviously there are a lot of ways of knowing about our environment, but that's clearly my background. And so what we've got here is an image from our report, and a few points I want to make about this one is firstly that we have quite a bit of data about our marine environment. It's usually collected for a specific purpose, such as consent monitoring or a particular isolated study. And so what you can see, I've given some examples of the types of data there, but one of the key things about that is that there's a lot of different data types there. So we don't, while we have a range of data, it doesn't tend to be consistent in terms of where it's collected or the methods by which it's collected. And that's because its purpose is a specific project, not broad state of the environment assessment. The other thing that we need to keep in mind about it is that it is all relatively recent. So we've only got really substantial data sets, at least that were available to me, from around about the 70s. So that's a relatively short time period when you consider how long we've had people acting in the environment on the land and in the sea in ways that affect environmental health. So this is an image of Picton from about the 1920s, and there are a lot of things that are potentially affecting human health that we can see there. There's vessel traffic associated with some of the fisheries. There's obviously been the building of infrastructure and roads, and if you look up in the hills there, you can see substantial deforestation, and that's caused a high degree of sediment input into our marine environment, but we really, at this stage, don't quite know how much. So a big problem in marine, in all assessments of environmental health, is that we actually don't have baseline information. We don't know what things were like before humans started changing the environment. Now, in terms of our report, we attempted to look at four main types of impact in the marine environment. So that was contamination, habitat integrity, biosecurity or invasive species, and fisheries decline. So today I'm just going to talk to you about some aspects of the first two of those stresses. So firstly, contamination. It's an extremely broad term. It can refer to any number of compounds or materials that enter the marine environment and cause some kinds of issues. So it can include sediment, it can include litter. Today I'm just going to talk about some toxic chemicals and organic matter as far as what we know of contamination. So one of the key sources of toxic chemical contamination that's relevant to Picton Bays is the antifouling compounds, and these are a range of products that are all used to stop this, which we similarly don't want. Obviously, fouling on vessels has problems in terms of vessel efficiency. It's also a bad thing in terms of biosecurity, so we need to control those. But the compounds that have been used are applied in paints to boats' hulls, and they tend to enter the marine environment either by leaching or by particles coming off perhaps during hull washing and being incorporated into sediments. These can be really quite persistent, and the data that we have does show some heavy contamination, particularly the Carey's Boatyard site, which I'm sure you're all familiar with. It was, I think, operating for about 100 years. And we can see there that there's still a profound footprint in that space. A couple of pieces of good news around that are that as you move away from the immediate area, you see a really rapid decline in those contaminant levels within the space of tens of metres. The other good thing in terms of environmental health into the future is that a lot of those compounds are no longer able to be used either at all or the way that they were used in the past. So in terms of what's going into the environment, inputs there have been reduced. Second sort of key source of chemical contaminant or toxic contaminants is stormwater, and this tends to be quite well monitored as part of the consents allowing those discharges. So we do see some elevation of some contaminants at point, but an interesting point with this monitoring is because we're monitoring near the discharge point and an increasing distance away, we're expecting to see a decline away from the stormwater point. Often we don't actually see that. So the monitoring associated with the stormwater inputs is actually identifying other sources of contamination that we're not necessarily entirely sure where they've come from. They may be historic, or they may be just coming from an unidentified source. So that's all I'm going to talk about as far as toxicants in the marine environment at this stage. I'd like to talk to you now about organic contamination of the marine environment. So I'm sure this is familiar to some of you. This is Shakespeare Bay, and you can see the outfall from the freezing works there. So I think the image will have been taken at some point in the 70s when freezing works discharges, including entire guts of cows, were discharged into the marine environment. As you can see, there's a rather choice quote there from the chief engineer to the, it's from the chief engineer to the chairman of the Marlborough Catchment Board. It says, "Inner Shakespeare Bay was discoloured a deep red brown, due mainly to blood and particles of fat, meat and animal faeces. The discoloured water was overlain by a thick transparent layer of fat." So it was an extremely heavily impacted site. There was a dead zone in the middle of the bay where not even these opportunistic pollution-tolerant species would live. There were sulphide gases discharged spontaneously from the seabed. It couldn't have been a lot worse, really. So clearly we no longer treat our marine environment in this manner. And work that happens now for a range of reasons in Shakespeare Bay shows that that seabed has greatly improved from the condition it was in sort of at the end of the 70s when the freezing work discharges finished. So the second key organic inputs came from sewage discharge, which was similarly untreated, discharged into the marine environment. What you can see here is a couple of sediment cores taken, I presume, by some diver, and I'm so pleased it wasn't me, near the outfall in the early 90s. So this is about 6 centimetre wide sediment core, and you can see that about the top 10 centimetres of that is completely black. So that means it's anoxic. There's been so much biological activity in there, all the oxygen's been used up. And similarly, very little would be able to live in there. The bacterial levels associated with that kind of discharge and with the Shakespeare Bay freezing works were extremely high, thousands of times what we would consider acceptable from a human health perspective, let alone from a cultural perspective. And now, we're obviously no longer discharging raw sewage into the environment in the manner that we were, and what we would see around the area now the second cores are not from the exact same site, but that's a more healthy environment. It's grey sediments rather than black, so it's oxygenated, and we're going to see that the communities in that are far less damaged than they were in the past. Now, the improvement in organic and bacterial contamination in the area shows up in council data. So this is recreational water quality data from council, and you can see the 5-year moving average in Picton has steadily declined over the years. It's still higher than ideal, but there are a lot of different sources that could come from, including farming, seabirds, boat discharges. There's a range of ways that we can still have bacterial fecal contamination of the marine environment, but there's been a great improvement there. Now, moving on to habitat integrity, and by that I mean the physical structure that supports natural communities. So that can be rocks or sediment, or it could be organisms such as seaweeds or shellfish beds that have a particular community associated with them. So I'm going to talk about the coastal margin, and then I'm going to talk briefly about the seabed. So what we have in this slide is an image of modern day Shakespeare Bay on the right. So we can see that there's been a bit of reclamation with the Waimahara Wharf and the log yards, but the important thing to see there is that we've got these intertidal sand flats remaining, and in this image here you can see the proposal to actually reclaim that entire area. Now, those intertidal sand flats are far more rare in Queen Charlotte Sound than the environments up the sides of the sounds, and they contain really important communities. So we can see from a recent photo that those sand flats support large seagrass beds. Now, these are important in terms of they're productive, they provide habitat for a range of fish and other animals, and also they mitigate the effects of certain activities on the land as that water flows into the marine environment. So it's great that that exists. We don't have any of that habitat surviving in Picton Harbour. You can see here the Waitohi Lagoon, which is obviously now the ferry wharves, and the Kaiawa Lagoon, which is now the marina. So those sand flats and estuarine habitats don't survive in that environment. And finally, in Waikawa, we do have small remnant sand flats here, and good news about those is that they are currently, or just have been surveyed as part of the Council's estuarine state of the environment monitoring, so we look forward to the information about those becoming available. So the last aspect of habitat integrity I want to talk about is firstly so it's the habitat integrity of the seabed. I want to talk to you about these images on the left, which are soft sediment habitats. Now, these images aren't actually taken locally, they're from Tasman Bay, but they show you a really common dynamic to do with disturbance of the seabed. So the top image is of a seabed that's exposed to disturbance in the form of bottom trawling and so on. And you can see that they're really fine sediments, and those burrows are formed by mobile animals that can recolonise after the seabed has been disturbed. The bottom image is actually under a mussel farm, and while you can see that there are some mussel shells there, there's a lot of other shells as well. And what we've got there is a much more stable environment. Those mussel shells can limit resuspension of the finer sediments, they create diversity, and they can provide a settlement habitat as well. So there are obvious sources of seabed disturbance in Picton Harbours, and in this kind of dynamic is what we expect to see. On the right, we have a rocky reef habitat. That is a local photo and characteristic of what we see around here. So we know, and I think Sean will speak some more about this, about that there used to be seaweed forests all along these rocky reefs, and a common dynamic that we see is when those larger predators are fished out, we see an increase in herbivores that can prey down the algae forest, so that takes away all that diverse structure which there was another whole community associated with. So what we've got there is quite a depauperate community, a little bit of pink encrusting algae, a little bit of green, fast-growing algae, and quite a few urchins, which they seem to be hiding under the algae or eating it or something here. Now, I've quite clearly stated this is oversimplified assessment of the state of the environment, but I've just tried to roughly put a traffic light sort of a indication of where we're at in terms of the state of particular stresses on the environment, what trend we're seeing, and what our level of data quality is on that. Purple indicates that something is really unknown. So you can see in terms of sediment input, we don't have a lot of information regarding what's happened in the past. We lack the baseline of change. Similarly, we're not quite sure. There's probably less sediment going in at the moment, but who knows? And so the data quality is quite poor. In terms of the toxic chemical contamination, there are good things happening in terms of some of those really toxic chemicals no longer being used, but there's still quite a bit of uncertainty around that. The organic matter contamination and the bacterial contamination, we're still obviously not pristine, it's not at baseline levels. However, there is a positive trend on the scale of the last 30, 40 years. Similarly, habitat integrity, the data quality is pretty good. Loss of habitat integrity from disturbance, again, the state and trend really aren't known because the data quality is fairly poor. If we do want to know more about this, there are things we can do for every component that I'm touching on there, and these are just examples. But in terms of the history of sedimentation, coring studies such as that currently underway in Pelorus can inform that kind of historical, you can recreate the historical baseline. It's possible to do more integrated broad-scale assessments if we do want to know more about the patterns of chemical contamination. As far as the organic matter contamination, it seems to me that that would be a good candidate for better integration across data sets, that there's quite a lot of information there, but it's collected for different purposes, so we can't quite line it up. In terms of the bacterial contamination, one of the things you can do is use molecular techniques, so you can actually look at the genetics of the bacteria and work out what kind of organism that came from, so that helps you track down what your sources are. Changes in habitat integrity as far as construction are pretty well documented nowadays, but the loss of habitat integrity associated with disturbance could be better informed by disturbance mapping, by the coring, and by local knowledge. So nearly finishing up here, but I just wanted to touch on the different kinds of information. So as I said, I'm coming at it from a Western science perspective, so that's got a bit of a patchy history. We're getting more data, but it's quite recent. It's not going to take us a long way back in time. Overall, there are some ways we can look back in time. Compared that to that M was me, so that's one individual's knowledge of the environment. But if we look at living individuals generally, and we were very lucky to have a conversation with Tom as part of this, Tom Norton as part of these projects, there's a lot of really informative information that locals hold about the environment. And then of course we need to recognise the knowledge of tangata whenua that extends far back beyond individual lifespans. So I think there's a really positive move towards integration of different kinds of data, and MDC have been doing some of that in their estuary monitoring and a number of other projects, and I just commend that. I just wanted to recognise Te Atiawa's assistance with our reporting, and they permitted us to use some of their iwi environmental management plan in our report, and I would just recommend the document to people in terms of the statement of aspirations and plans for improved environmental management is really quite impressive, and I just wanted to acknowledge their permitting us to use some of that material. Also, apologies, the text has gone a bit odd again. Thanks to the EnviroLink program for funding our reports, to MDC for involving us in the work, and my colleagues at Cawthron, Liv Johnston and Paul Gillespie, for their involvement in my report. So I'm not sure if I've gone over time, but thank you very much, and if we have time for questions, please let me know.
That's fascinating. Thank you very much, Emma.